Here is what keeps peptide chemists up at night. You can build a peptide with the exact right sequence, confirm the exact right mass, and still end up with a drug that doesn't work. Same atoms, same mass, wrong medicine. The culprit is often two sulfur atoms that found the wrong partner. One disulfide bond out of place. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams to characterize peptide drugs and prove their disulfide bonds sit exactly where they should. Today, we'll unpack how one wrong disulfide bond can quietly ruin your peptide drug and how the lab catches it. If you have found anything valuable in this channel and videos, please like this video and subscribe our channel. This gesture of appreciation means a lot to us. Let's start with the molecule that taught the industry this lesson: insulin. Insulin holds its active shape with 3 disulfide bonds, and getting them to pair correctly took decades. Amphisen showed that a peptide sequence carries the instructions for its own folding. The disulfides lock the shape that makes the drug work. When recombinant insulin arrived in 1980s, the hard part wasn't building the chain, it was getting those 3 bonds to pair right. Right sequence, wrong bonds, and you don't have insulin, you have an inactive look-alike. So, what is a disulfide bond? It's a covalent link between 2 cysteine amino acids, 2 sulfur atoms joining to form a cysteine bridge. Think of these bridges as the rivets that hold a peptide shape together. They are everywhere. Insulin with its 3 bonds, oxytocin where a single bond closes the ring, octotide and linaclotide stitched together by several. The bond isn't decoration, it's what makes the molecule fold into something a receptor will recognize. A disulfide bond can go wrong in a way that's almost invisible. Say a peptide has 4 cysteines, the correct drug pairs 1 with 3 and 2 with 4. But during folding, 1 and 2 can bond instead. Same peptide, same 4 sulfurs, completely different shape. That's a disulfide isomer, and here is the unsettling part. It has the identical formula and mass as the real drug. On a mass spectrometer, the intact molecule looks perfect. The number is right, the medicine is wrong, and free thiols or trisulfides where an extra sulfur sneaks in and you have a family of lookalikes, a simple mass check will never flag. A mispaired disulfide bond costs you in 3 concrete ways. First, potency. The wrong fold means the peptide no longer fits its receptor, so the drug simply stops working. You have made an inactive impurity. Second, safety. A misfolded variant can look foreign to the immune system and trigger an anti-drug antibodies, the immunogenicity risk every biologic team worries about. And third, regulatory. Those isomers are product-related impurities, and if you can't identify and control them, your filing stalls. 1 molecule, 3 failure modes, all tracing back to 2 sulfur atoms in the wrong place. Now, not every peptide carries the same risk. The more cysteines a molecule has, the more ways they can pair up wrong. A peptide with 3 disulfide bonds can misfold into dozens of isomers. 2 chain peptides like insulin add another layer because the chains must find each other correctly too. Even a single bond cyclic peptides can pop open and dimerize. So the first question is always how many cysteines you're dealing with. If you have watched a right mass, wrong result, finding derail a project. Tell me in the comments those stories say which breakdowns I make next. Consider 3 real examples every peptide team should know. Insulin again. Mispaired disulfide isomers are biologically inactive, so pharmacopoeias cap them as related substances. Second, recombinant human growth hormone can form a trisulfide variant, an extra sulfur wedged in during expression that has to be identified and controlled. And third, synthetic peptide generics. Since 2021, FDA guidance requires sponsors to characterize disulfide isomer impurities before an ANDA is approved. At Resolve Mass, we have helped sponsors chase one unexpected isomer peak that became a control strategy rewrite and delayed filing. The molecules differ, the lesson is identical. Disulfide connectivity is a critical quality attribute, not a footnote. So, how does the lab catch a wrong bond? You can't weigh your way out. The mass is identical, so you map the connectivity directly. You digest the peptide into fragments while the disulfide are still intact, then run a non-reduced peptide map on LCMS to see which pieces are linked. Compare that to a reduced map and the linkages reveal themselves. You check for free thiols, confirm potency, and this is the part teams underestimate. You keep the digestion cold and acidic. High pH scrambles disulfides during the sample prep and invents impurities that were never there. Your own method can lie to you. So here is what to walk away with. In a peptide drug, connectivity is the medicine, not just the sequence and not just the mass. Disulfide isomers share the exact same molecular weight of the real thing, so only connectivity mapping can find them. A single mispaired bond can cost you potency, patient safety, and approval. Map those bonds early and digest cold so your own method doesn't create the problem. If you're developing a peptide, an IND, an ANDA, or a biologic, and you want a CDMO or CRO partner who treats disulfide connectivity as the critical quality attribute rather than a box to tick, Resolve Mass Laboratories supports small and mid-sized team from early characterization all the way through submission. Thank you so much for watching this video till the end. Before you go, I want you to do 3 quick things. First, subscribe to this channel right now. The more you subscribe, the more valuable pharma insights, expert tips, and industry updates we can bring to you. Please comment and let us know what did we do wrong and what did we do right in this video, because the more you give us the feedback, the better we will be able to make videos for you. And share this video with at least one person who will find it useful. You never know who is really stuck in this kind of problem right now. I will see you in the next one. Until then, goodbye for now.